Good afternoon, good morning, good evening to everyone. We welcome you all today, particularly those of you joining from far away or at very late or early times. Welcome today, and thank you for joining this WHO EPI-WIN webinar on pathogen genomics in health emergencies, Spotlight Dengue. We're hosting this webinar in collaboration with the International Pathogen Surveillance Network. I'm Supriya Besbarwa, lead for EPI-WIN, along with my colleague Sudrish Pradhan here. For those of you new to EPI-WIN, this is the WHO Information Network for Epidemics for Science and Knowledge Translation in Health Emergencies. With these EPI-WIN webinars and the associated updates, our goal is to translate science in an accessible, understandable, and relevant way so that we can have evidence-informed actions and decisions in public health emergency preparedness and response. And today we have a really great panel from whom we will hear about pathogen genomic tools and how it is being used in dengue outbreaks. We have giving the opening remarks, Dr. Josefina Campos, who's the unit head for genomics and analytics in WHO Health Emergencies. Then we have very eminent panelists. We have Professor Nuna Faria. He's a professor in virus genomic epidemiology at the Faculty of Medicine in Imperial College London in the UK. We have Dr. Gamu Fall, who's joining us in spite of being very busy also with Mpox. He is from the WHO Collaborating Center for Arbovirus and Hemorrhagic Fever at Institute Pasteur in Dakar, Senegal. And finally, last but definitely not least, Professor Li Ching, Group Director of Environmental Health Institute at the National Environmental Agency in Singapore. We also have with us Dr. Diana Rojas Alvarez, who will provide the closing remarks. She's the team lead for iboviruses in WHO and a familiar face at EPI-WIN. And before we go on to the main webinar, I'd like to give you some housekeeping announcement. First of all, as those of you familiar already know, this webinar is being live streamed to YouTube. It's at the EPI-WIN YouTube channel, if you'd like to go there. The webinar is also being recorded, and afterwards the recording will be available from our website, and we will share it also with everyone who registered. We are having interpretation today in French and Spanish, so please go to the interpretation globe icon to access and select the language that you want the interpretation in. We invite you to post your name, location, and organization. EPI-WIN is EPI-WIN because of all of you, so we'd love to hear from you and where you're from. Do tell us your name, location, and organization in the chat box. But please put your questions in the Q&A box. This is important because if you put your questions in the chat box, we might sometimes miss it because there's so many comments and all of that. So for us to see your question as a question, we request you to put it in the Q&A box. And before I hand the floor over to Josefina, we would like to hear from you. I request my colleague Sadrish to share a Slido. Very quick question. Do you work in the area of genomics? Please scan the QR code or click on the link and answer. If you go scan the QR code on the screen or join us at slido.com, hashtag 1467766. So we just wait a few minutes to hear from you. We'd love to know about you. So please either scan the QR code on the screen or go to slido.com and hashtag 1467766. Okay, it looks like, well, 60% no. The number of yeses is going up as we have more and more participants. Now it's 57-43. Right? That's great to know. Thank you very much. And with this, I hand over to Josephina. Over to you, Josephina, to go forward with the technical part of the webinar.
Thank you, Supriya. So good morning, good evening, good afternoon to everyone. So today we are having this webinar on behalf of the International Pathogen Surveillance Network, is IPSN, to tell you a little bit about this initiative. It's a global network of pathogen genomic actors that it was brought together by the WHO Hub for Pandemic and Epidemic Intelligence. And the aim of this network is to accelerate the progress in pathogen genomics to improve public health decision making, acknowledging all the progress that countries have been done to respond to SARS-CoV-2 pandemic. This network provides support to public health community in how to best leverage the power of genomics in their pathogen surveillance and response activity. And with that idea of informing country capacity building activities, we want to today kick off a series of webinars on pathogen genomics in health emergency, with the aim to provide a forum for experts and evidence-based discussions on the role of genomics in the response to ongoing outbreaks, considering where and where not to use genomics and where can assist in public health decision-making for emergency. In this first webinar, co-hosted with the WHO Avovirus team, we want to spotlight the use of genomics in supporting the response to the current dengue outbreak and its role in strengthening the overall surveillance for public health interventions. For this, we got together speakers from country perspective and academic perspective from Imperial College London, National Environment Agency in Singapore, and Institut Pasteur Dakar in Senegal. And with that, I will give the floor to Professor Nuno Farias to start with his presentation. Please, Nuno, the floor is yours.
Thank you very much, Josephina. Let me just share the slides. Can you see my slides?
Yes, we can, Nuno. Over to you.
All right, fantastic. Good morning, everyone. Good afternoon, everyone. I'm Professor Nuno Faria from Imperial College, but also from the University of São Paulo. And today I'll be discussing the role of genomics in responding to dengue health emergencies, a topic that has become increasingly important as we witness the rapid increase of dengue worldwide. As of August 2024, nearly 11.5 million dengue cases and more than 7,000 dengue-related deaths have been reported to WHO from all regions across the globe. Of these, 9 million were reported in Brazil, that experienced its largest ever dengue outbreak. But active transmission of dengue is still occurring in over 90 countries across all WHO regions. There's a significant increase in the number, in the scale, and the simultaneous occurrence of multiple outbreaks across different regions. So that demonstrates the pervasive and the growing threat of dengue globally. Dengue is a viral infection that is transmitted to humans through the bite of infected mosquitoes, mainly Aedes aegypti and sometimes Aedes albopictus. Dengue is predominantly transmitted in an urban cycle, where the virus is transmitted between humans via an Aedes mosquito in urban and semi-urban areas. In some areas of Southeast Asia and West Africa, sylvatic dengue lineages occasionally jump to humans, causing small outbreaks. How does transmission primarily occur? So first, a mosquito feeds on an infected person. Dengue virus then infects the mosquito's midgut cells, and within a week or so, it spreads to the mosquito's salivary glands. Once infected, a single mosquito can transmit the virus to several humans during its short lifetime, which is about 2 weeks. In humans, after being bitten by an infected mosquito, symptoms may appear up to 4 to 7 days, and about 20 to 80% of infected humans are symptomatic, meaning no symptoms, but they can still contribute to transmission. The rise in global dengue epidemics is closely linked to expanding range of these mosquitoes, Aedes mosquitoes, which have now invaded tropical and subtropical areas, and more recently also temperate areas both in the northern and southern hemisphere. This expansion is mainly driven by warmer climates, which, coupled with rapid urbanization, increased population movement, fragile health systems, and inadequate waste management in urban settings, can create the conditions for large dengue outbreaks. And more recent modeling estimates shows that 5.7 billion people, that is 7 out of the 10 of us, are at risk of infection with dengue. So dengue is part of the flavivirus genus, which also includes yellow fever, West Nile, and Zika viruses. Dengue can be classified into 4 genetically divergent and antigenically distinct serotypes, which are called dengue-1 serotypes 1 to 4. So each serotype has a broad genetic diversity and can be further classified into genotypes. I will talk more about this in a few minutes. Some serotypes and genotypes have been associated with increased disease severity. For example, in the lower plot here, showed that, a recent study showed that dengue-3 in Nicaragua leads to more severe outcomes in both primary and secondary infections. But infection with all serotypes can cause high fever, headache, body ache, nausea, and rash. However, in severe cases, disease can be fatal, as it can lead to plasma leakage, severe bleeding, and organ impairment. Following a dengue infection, neutralizing antibodies, following a dengue infection, neutralizing antibodies at high titers are observed to provide long-lasting protection against the infecting serotype. However, dengue infection also elicits low to intermediate cross-reactive antibody titers, which can increase the risk of a symptomatic infection and enhance disease severity in a subsequent dengue infection with a different serotype. This is a phenomenon known as antibody-dependent enhancement. Sequential dengue infections with different serotypes, and therefore increased risk of severe dengue, is facilitated by the co-circulation of multiple serotypes. Several serotypes are now endemic in different regions across the globe, as shown here on the right-hand side. Given dengue global distribution, escalating epidemics due to co-circulation of serotypes, and the potential for causing severe outbreaks, dengue virus is now being classified by WHO as a priority pathogen. Laboratory surveillance plays a fundamental role on the global response to dengue. The most commonly used diagnostic approaches involve the direct detection of either particular regions in the dengue genome via RT-PCR or viral proteins such as NS1 using antigen tests. These methods can be used to detect dengue acute infections, which is typically 1 week at most after symptom onset. In contrast, serological methods detect the antibody response generated against viral infection, particularly IgG and IgM antibodies, and less often neutralizing antibodies using a technique called PRNT. Other diagnostic methods, including LAMP-based or CRISP-based diagnostics that do not require cold chain, and untargeted metagenomic sequencing, are still mostly investigational, but have shown great promise in arboviral diagnostics. But despite recent advances, there are still disparities in surveillance capacity at regional and national levels, with several important gaps in arbovirus surveillance globally, which are shown in the darker blue areas on the map on the right-hand side. However, several strategies can help to reduce gaps in surveillance. So this includes investing in laboratory infrastructure and capacity strengthening initiatives, expanding access to diagnostics, improving awareness and healthcare management of dengue, and ensuring sustainable funding and resources for dengue surveillance programs. Dengue is an enveloped, positive sense and single-stranded RNA virus with about a genome which has about 10.7 thousand base pairs. Its diameter is around 50 nanometers, and that is around 2,000 times thinner than a human hair. Its genome structure, shown here on the upper right side, contains a single opening reading frame that encodes 3 structural proteins. This includes the envelope protein that the virus uses to attach itself to host cells, and several non-structural proteins, which includes NS1 detected in antigen tests. As a typical RNA virus, dengue virus genomes evolve rapidly. This is due to a combination of high mutation rates, large population sizes during acute infections, and short generation times, with new variants typically generated within days. Its evolutionary rate is around 10 to the minus 3 substitutions per site per year, which corresponds to 10 nucleotide changes becoming fixed at the population level per year. This is actually quite similar, for example, to the evolutionary rate of SARS-CoV-2. The phylogenetic tree of dengue on the right-hand side below illustrates this. Each tip corresponds to a sequenced virus, and tips are colored according to the date of sample collection. Horizontal lines correspond to phylogenetic branches that are scaled in number of nucleotide changes. And what is clear from this is that contemporaneous dengue sequences, in dark blue, are significantly different to those circulating in the previous decades, with over 300 mutations separating them from viruses circulating in the 1980s. This rapid evolution facilitates the potential emergence of novel strains that can evade host immune responses and brings additional challenges for diagnostics and vaccine development. During the COVID pandemic, genomic sequencing was critical to detect the emergence of novel lineages associated with faster transmissibility and more severe outcomes, and plan, of course, data-driven interventions. SARS-CoV-2 has been by far the most sequenced pathogen, with over 16 million genomes shared from nearly every country in the world. So how does this compare to other pathogenic threats? So this plot shows the number of sequences in a log scale currently available for 18 highly pathogenic viruses, the ones causing highest number of deaths after the 1980 influenza pandemic. Red boxes below indicate public health emergencies of international concern. But what I want you to show here is that dengue is currently the most sequenced arbovirus, followed by West Nile, chikungunya, Zika, and Venezuelan equine encephalitis virus, and is overall the 5th most sequenced viral threat. Up until today, more specifically, 43,000 dengue sequences have been shared in public databases. 90% of these are partial genomes, and 10% of these are whole genomes. So the plot on the left shows a proportion of dengue serotypes per WHO region. And this data shows that dengue 1 and dengue 2 sequences predominate across all WHO regions, while dengue 4 seems to be less dominant serotype with rare or no circulation in Europe and the Eastern Mediterranean region, and few detections in Africa and Western Pacific regions. In Europe, Aedes albopictus has now established populations in 13 different countries, leading to sporadic dengue outbreaks. Since 2010, Europe has reported 285 locally acquired dengue cases, with recent localized outbreaks detected in Italy and France just this month. In the Western Pacific regions, Southeast Asia and the America regions, all serotypes circulate, with an apparent increase in dengue 1 and 2 detections in the Americas. These regions have shared over 8,000 sequences each. And within the Americas, the recent dengue epidemic in Brazil was caused by co-circulation of dengue 1, genotype 5, and dengue 2, genotype 2, although dengue 3 has also been recently detected. Traditionally, the classification of dengue genetic diversity has involved classifying strains at the serotype and also genotype level. Different teams across the world would then provide additional and often different classification to clades within different genotypes. So in an attempt to standardize dengue lineage classification nomenclature, recently, 2 subgenotypic levels of dengue virus classification have been proposed, major and minor lineages. So let's look, for example, at serotype 3, shown here. This serotype can be classified into 4 genotypes, and according to this new classification system, 12 major lineages and 13 minor lineages. The expectation is that more granular nomenclature systems, such as this one for dengue virus, will allow to better identify and track lineages of potential epidemiological and/or clinical importance. But how can we actually generate dengue virus sequences? First, if available, we need to adapt existing sequencing capacity and frameworks built during the COVID-19 pandemic. Second, provided that there is access to PCR-positive samples and linked metadata, one can select specific samples for genome sequencing according to a particular sampling strategy. After nucleic acid extraction, virus amplification, and sequencing library preparation, genomic data is generated using next-generation sequencing technologies. For sequencing SARS-CoV-2, for example, Illumina and Oxford Nanopore were the most frequently used technologies. The next step involves using standardized bioinformatic pipelines to process raw sequencing data, perform quality control, and generate consensus sequences. Sequences can be shared using several platforms, including NCBI, GenBank, and GS8. To reconstruct transmission patterns from genome sequences, we need to integrate new genomes with reference or background genomic datasets and epidemiological data. This is really important. Epidemiological data, for example, can include patients' travel history, age, sex, and clinical outcomes. Using phylogenetic approaches, we can then track and monitor the spread of particular lineages and identify mutations with potential phenotypic impact, for example. Appropriate data interpretation and translation to outbreak responders is critical to maximize the public health impact of sequencing programs. This can be facilitated through sharing results with key stakeholders and with the community through, for example, preprints via dashboards such as NextTrain or MicroReact, preferably in combination with diagnostic and entomological surveillance data. So now I'm going to show you 6 examples on how genomics is impacting dengue public health. First, dengue genomics allows investigating serotype, genotype, and lineage transmission and spread at different scales. For example, the phylogenetic tree reconstruction here, shown in the video, highlights how over more recent decades, the pace of dengue virus serotypes increased significantly due to increases in air travel volumes across the world. At a much smaller spatial scale, the study on the right used virus sequences from Bangkok City in Thailand to show that sequential transmission of dengue during the same season typically occurs between households and in the same neighborhood. However, across different seasons in the lower panel, viral lineages become well mixed across Thailand, both within Bangkok and between different provinces in Thailand. So this shows that dengue epidemics in Bangkok are driven by introductions and subsequent spread of dengue in the city, which later acts as a source of viral lineages that are transported elsewhere. Pass. Okay. Sequencing augments traditional surveillance approaches and is particularly useful to identify local circulation of dengue lineages. The study on the left, for example, analyzed sequences from the emerging dengue tree genotype tree in the USA. This study used analysis of sequencing data to show that most locally acquired cases occurred shortly after introduction, with little evidence of sustained transmission. Infectious disease surveillance, however, on the right-hand side of travelers can also supplement local surveillance. Infected travelers can be considered as sort of sentinels for pathogen transmission in locations where outbreaks have not yet been reported. So the example on the right-hand side is based on genome sequencing of viruses from travelers detected in the US, but infected in several Caribbean countries. And this study provided evidence of dengue tree, genotype tree, cryptic or silent circulation in the Caribbean before its introduction in the US, where it eventually led to locally acquired infections in Florida. Dengue genomics has also been essential in the development of vaccines, and to date, 2 dengue vaccines have been licensed, Denguevaxia and Qdenga. Another promising vaccine is the single-dose dengue Butantan vaccine developed by the Butantan Institute, which is in the late stages of clinical development and will likely be licensed by next year. Denguevaxia, however, was the first dengue vaccine to be licensed and is currently not being used widely. It is a live recombinant tetravalent dengue vaccine, which uses yellow fever virus backbone and structural genes from dengue 1 to 4 genotypes, shown here on the right, in the bottom with double crosses. Analysis of data from 2 trials conducted across 10 countries show that dengue vaccine efficacy is lower against strains from different genotypes than those used to manufacture the vaccine. So the take-home message here is that dengue genomics is essential to guarantee that we can generate effective vaccines that confer protection against the evolving landscape of dengue viruses. Sequencing is also essential to improve molecular diagnostics, particularly in underrepresented settings. In 2023, African CDC reported 18 African Union member states reported outbreaks of dengue, among which 7 had ongoing armed conflicts. Aedes aegypti has been reported in 47 out of 55 African Union member states, but nevertheless, only 34 of the 7 countries with the presence of Aedes vector have reported cases of dengue so far. Genome sequences from Africa correspond to around 4% of total dengue sequences available, and this study generated novel dengue genomes from Burkina Faso, where limited data had been available until then. The authors found several mutations in dengue virus gene targets in strains circulating in Burkina Faso, and importantly, they found that mismatches between primers and/or RT-PCR probes and binding sites are pervasive, particularly for dengue 2 and for Africa, which may result in false negatives or inconclusive results. Phylogenetic and phylodynamic approaches can uncover information in sample genomes and are often analyzed together with other data sources, such as climate, demographic, and spatial data. On the left, phylodynamic methods were used to estimate from sequence data alone dengue effective reproduction numbers, defined as the average number of secondary infections produced by a single infected individual in the Hunan province in China. So the blue bars here correspond to winter periods. And using genetic data, the study found an effective reproduction number below 1 during winter, and therefore no evidence of local persistence of dengue lineages throughout the year. Instead, the annual epidemics in Hunan were driven by introductions from neighboring countries. Moreover, on the right-hand side, phylogeographic models can also be used to explore the contribution of different factors potentially associated with dengue spread at different scales. These models have often shown that air travel volume played a critical role in the spread of dengue, both within countries and within regions. Finally, strengthening genomic surveillance for dengue will also contribute to improve pandemic preparedness against pathogen X, which is a placeholder to define a pandemic pathogen that has not yet been characterized. Targeted metagenomics, which is the process of sequencing the total nucleic acid content in a sample, allows the genomic characterization of known and novel viruses in an untargeted manner. So this method has been used to detect undiagnosed dengue infections, for example, in Senegal and Uganda, but also to detect novel or rare viruses. In Brazil, metagenomic sequencing was performed on samples from 8 fatal cases in São Paulo. In 2 of these patients, a highly pathogenic arenavirus called Sabia virus was detected. Ribavirin has been successfully used to treat lab-based acquired infections with Sabia virus in the past. So looking ahead, the implementation of rapid metagenomic sequencing in clinical settings holds the potential to significantly improve patient outcomes and potentially to save lives. So to summarize, dengue is a global health priority, and genomic surveillance is essential for understanding its transmission, improving diagnostics, and guiding vaccine development. Strengthening global collaboration in these areas will improve our capacity to respond to dengue and other emerging viral threats. And looking forward, we must enhance genomic-based surveillance to improve detection, prediction, and response to dengue and other viral threats. So this includes using representative genomic data to optimize future vaccine design and strengthen collaborative surveillance across disease and threat surveillance systems, across sectors, across emergency cycles, and across geographical regions. Thank you to all the participants and to WHO for organizing this seminar.
You are on mute, Josefina.
Thanks very much, Nuno. So we have some questions from the audience, but we will go to our next participants and we have the questions at the end. Dr. Gamou from Institut Pasteur in Dakar, please, the floor is yours.
Yes, thank you very much. Can you see my slides?
Yes, perfectly.
Okay, thank you very much. So I'm Dr. Gamou Fall. I'm a virologist working at the WHO Collaborating Center for Arbovirus and Hemorrhagic Fever Viruses at Pasteur Institute in Senegal. And I'm going to present the role of genomics in the response to dengue in Africa, particularly in Senegal. So this is the global dengue situation that was already presented by Professor Faria. Now coming to the Africa region. In 2023, by end of December, about 170,000 cases of dengue were reported, and this was associated with about 750 deaths. And the outbreaks were reported in 15 countries, and the most affected one was Burkina Faso, that registered about 68,000 of probable cases. It's probable because the case were just confirmed by a rapid diagnostic test. And this outbreak was associated with about 680 deaths. And this is the situation in the Africa region in 2024 from January to end of August. So we have currently about 20,000 confirmed cases and 1,700 severe cases and about 71 deaths. And again, we can see that about 7,000 of the confirmed cases were detected in Burkina Faso. So in Africa, despite ongoing efforts in many countries, the dengue epidemiology is still not very understood. And this is due to many reasons, including the similarity of the symptoms between dengue and other tropical disease like malaria. It is also due to the limited laboratory capacity to timely detect and confirm dengue cases, and this is very crucial for rapid response to prevent disease spread. And also in many countries, we don't have appropriate surveillance systems for the dengue detection. And these are discussed in some publications, raising the need to have real-time robust dengue surveillance in Africa. I'm struggling with the slides. Can you still see my slides?
Yes, we can see.
Yeah, okay, but I cannot move anymore. I don't know why. Excuse me. Okay, sorry. With regards to this lack of surveillance systems in Africa, what is very clear is that in many countries, we don't have dengue-specific surveillance program. And usually in some countries, dengue is tested through the differential diagnosis of yellow fever because we have this yellow fever lab network, which is very well organized in many countries, and particularly in Senegal, dengue is routinely tested on all negative yellow fever samples. In Senegal, we also have a surveillance in hospitals of viral hemorrhagic fever cases. And again, in the differential diagnosis, dengue is also tested. We also have a surveillance system in Senegal in the southeast of the country, in the region called Kédougou. Kédougou is an area with very big forest, with non-human primates, and the environment is really favorable for arbovirus circulation, including dengue, chikungunya, Zika, and yellow fever. And we have a lot of gold mining activities, so a lot of people are coming from other countries to Kédougou for these gold mining activities. And we have a surveillance there that is done in collaboration with the Ministry of Health and funded by the NIH since 2009. And with all these different systems, I can tell you that the dengue detection was really rare. Just a few cases were detected in the country. But from 2015, the Ministry of Health, in collaboration with Institut Pasteur de Dakar, have implemented Sentinel syndromic surveillance, in the country. And this one is using a case definition that is quite broad. It's a fever associated with at least 2 of the following signs. It can be headache, arthralgia, rash, jaundice, and so on. And we have at least 1 sentinel site in each region of the country. And currently we have more than 30 sentinel sites across the country. And in each sentinel site, healthcare workers are collecting information regarding the symptoms and the epidemiological data of the patients. And on a daily basis, they are sharing these information using their Android tablets through an application that is developed by Institut Pasteur de Dakar. So we can analyze on a daily basis those information and generate these kind of peaks. And whenever the peaks are beyond the threshold, it means that something is happening in that area, and usually it's an outbreak. And in parallel, they are also shipping blood samples to the laboratory for arbovirus testing, including dengue. And for the testing, we are using different methods. As Professor Faria said, RT-PCR, NS1 antigen testing, ELISA, and PRNT to confirm, as we have other flaviviruses circulating in the country. And we are also doing viral isolation and sequencing. So for the sequencing, this is the workflow that we usually used. So samples are collected in the field and shipped to the laboratory. So for dengue, we first test for a pan-dengue detection. And once we have positive samples, we perform the serotyping using PCR kits. And after that, we perform viral genome sequencing to be able to perform phylogenetic and temporal and spatial distribution of the dengue viruses. And here you can see all the devices that we have at IPD for viral genome sequencing. And with all those systems, and particularly the Sentinel surveillance, I will show you now the evolution of dengue detection. So first of all, this is the first dengue cases that we had in the country in 1970. And the first urban outbreak in Senegal was detected in 2009. And from there to now, we have every year now dengue outbreaks. And currently we have multifoci outbreaks and multiple serotypes circulating at the same time. And here you can see that the 4S network is kind of early warning system, very effective for arbovirus detection, including dengue, because usually the patients are samples at the early stage of the disease, and this allows the laboratory to detect the case by RT-PCR, so we can be able to perform viral genome sequencing and perform all the analysis that are needed to better understand the outbreak. And this also gives the opportunity to the healthcare workers to better manage the patients. And here you can see the dengue evolution with the implementation of this sentinel surveillance. So from 2015, the detection was quite rare. And here you can see now all these dots in different areas showing that dengue is everywhere in the country. So this is from 2015 to 2021. And here from 2021 to last year. And we even detected an area with atypical dengue circulation, very huge circulation of dengue, and atypical in the sense of the timing of the detection, because usually the virus is detected in September, December, or early January in humans. But in this area, until April, the detection was ongoing, and some investigations, of course, were done with the Ministry of Health to better understand what is going on in that area. So now, why we need to do genomics? I think Professor Faria already talked about that. It's very important to do genomics to better understand the circulation dynamics of the serotypes and the genotypes that are circulating. And here you can see the dynamics of circulation of the serotypes from 2019 to early 2023 in Senegal, where we can see genotype 1 here in pink and genotype 2 and 3. So we can see that the main genotype circulating is the dengue 3. But in late 2023, we had of course, dengue 3, but the serotype 1 came back again. So last summer, we had both of them circulating at the same time. And also last summer, we got some severe cases with hemorrhagic fever that was confirmed to be dengue cases. And this genomics allow us, of course, to identify all the genotypes. And here, focusing on dengue 3, we identified 3 different introductions of the virus from Asia and Burkina Faso to Senegal. Dengue genomics can also help us to better understand the viral spread between the regions and between African countries. And of course, to identify also importation events, like here in 2017, we detected dengue 2 cases from Cote d'Ivoire to Senegal. Dengue cases can also help, genomics, can also help to improve diagnostic tools. And here, between 2021 and 2022, we detected a particular dengue outbreak due to sylvatic dengue 2 in the southeast of Senegal, Kédougou, which I talked about before, that caused about 60 human cases. And when we try to do the serotyping of those samples, the kit we used failed to detect those viruses because of some mutations that are present in the sylvatic dengue and that are not in the epidemic normal dengue virus. So we developed new tools that allow us to detect the new sylvatic dengue and introduce them in the panel of serotyping to be able now to detect the sylvatic dengue among human cases that will occur in the country. And finally, we also compare dengue 3 strands with some vaccine strands. And of course, again, we identify some mutations that could impact the vaccine efficacy. So it's really important to perform genomics for dengue to be able to identify those mutations and to support vaccine development. So in conclusion, in Africa, we can say that we have increasing dengue incidence, particularly in West Africa, with viral dispersion between countries and between regions inside country. We have in many countries now multifoci and multiserotype dengue outbreaks, which give high probability for secondary case to occur. And we have also some mutations, high genetic diversity that can impair diagnostic or prevention tools. So it's really important for endemic countries to strengthen dengue surveillance and to have efficient diagnostic tools for rapid case detection and management. As I said, probably severe cases will occur in many countries due probably to secondary dengue infection because of these multiserotype outbreaks. I think having tools that will allow us to differentiate primary versus secondary dengue infection will be very, very helpful. And of course, performing virus genomic surveillance to identify and characterize the circulating strains will be also very helpful to improve diagnostic tools and support vaccine development, and of course, better understand also the transmission dynamics and viral spread of dengue. And all this call for an implementation in Africa of a network for genomic surveillance for the better understanding of and better management of dengue. So I would like to thank all the organizers and the participants. Thank you.
Thanks very much, Dr. Gamal, and give us the perspective of Senegal and the Africa view. And with that, I will give the floor to our last presenter, Professor Litin. The floor is yours to talk about molecular epidemiology of dengue in Singapore.
Hello, can you hear me? Yes?
Perfectly.
Okay, hi. Thanks WHO and partners for organizing this meeting and for the invitation to share Singapore's story. Dengue has been in Singapore for many decades. The first reported dengue outbreak was 1901. If That's even when dengue was known, the virus was known. So if you're interested to find out how they describe the disease even before the 4 serotype was known, you can contact me. I will send you the literature. So in Singapore, we do a lot of molecular epidemiology, looking at viruses, whether it's dengue, Zika, chikungunya, to help us with risk assessment and to help us control the virus. I think it's very important to do sequencing and associate them with the epidemiology and associate them with the clinical outcome so that we can understand epidemic potential and disease severity of dengue viruses. I think there's quite a few questions out there in the Q&A posing the same. And I think more work needs to be done. I will present what we have. In dengue, it's 3 guilty parties, right? We have mosquitoes, yes, as Nuno has mentioned, there's virus, and there's people. These 3 guilty parties make one dengue case. If you take away one, there'll be no dengue. Of course, you can't take away people. Virus is always circulating because of the globalization, and mosquito is where then your susceptibility becomes very dependent on whether the mosquito is present. So 3 guilty parties form a case. And this case, if you don't control this case, if you don't protect this case, and the mosquito bites him, the mosquito will transmit the virus to the people around this case. Then you form a cluster. And if you don't control the cluster, it becomes an epidemic. When you form more and more clusters through the country, then you can become an epidemic. So if you look at this flow, there are places, there are really checkpoints for opportunities for intervention. Number 1 is alert. When is outbreak coming? So an early alert is very important for preparedness. And in this kind of in this situation, we do a lot of risk assessment and both temporal and spatial risk assessment. And if there is a cluster, then it's about prompt intervention to mitigate the transmission. So this is Singapore's strategy. And in fact, we want to move earlier before to say, don't wait for the cluster. Let's have an accurate diagnostic and case notification. So it is compulsory in Singapore. It's mandatory for doctors and for the laboratories to notify Ministry of Health of any dengue cases. Now, if we don't want any dengue, if we want to just prevent dengue cases, then we got to go even more upstream, which is number 1, control the mosquito population. Yeah, you suppress the mosquito population through source reduction. In Singapore, we mainly do by environmental management, removing breeding habitats. And more recent years, we have introduced Project Wolbachia, using Wolbachia ADs to suppress mosquito population to complement environmental management. We are not going to talk about that tonight, though it's today. It's night for me today because it's a topic on its own. But I think understanding the virus, its epidemic potential is very important. We have very little understanding. Unlike COVID, unlike even chikungunya, dengue viruses is just it's a mess. It's so much mutation have gone through for decades that tracking it is now very difficult. Understanding immunity and epidemiology of people is very important too for us. We know that we have very low herd immunity. We've done a lot of seroprevalence. I'm not going to present here, but again, understanding the whole system, the epidemiology, the vector, the virus, the people, and the environmental factors are all critical for control of dengue. Today, I'm just going to focus a bit on the surveillance of viruses. This is our program. Surveillance of case, virus, vectors, and ecology forms one pillar, one very robust pillar, important pillar for us, on top of preventive control, outbreak management, and public communication. So each of them is a talk by itself. I will not dwell into it, but today I will talk about viruses. So this is Singapore's epidemiology. The blue lines represent the house index, the number of it should be house index. On the right side, it should be blue in color, yeah? The red line is the number of cases. In the since 1960s, we've introduced a lot of program, and including clearing of slums and building proper housing, public housing for the population. And different acts and different program has led to the plunge in the house index. House index is the number of homes found breeding among 100 check. Okay, but dengue it was on the rise. So two main factors here. One is that we have very low herd immunity, as evidenced by our seroprevalence data. And secondly, it's in fact improved surveillance. Through the years, more diagnostic kits have been developed, and we know that we have been pushing for surveillance, and some of these rises could be due to surveillance because we don't track just DHF anymore, which we used to do. Even mild cases of dengue fever are notified. Okay, but more importantly here, I would like to just highlight the rise and fall of dengue. It comes and goes. It's pretty unpredictable. When is dengue going to be a problem? Is it this year or is it next year? As we can demonstrate from this chart here, look at that 2019, 2000, and all the years. You got some low years and some high years. So what caused all these up and down? One factor that we have realized since 2004, 2005 was a switch in the predominant serotype. That's in Singapore. I think in different setting, different epidemiological setting, you need to test this in your context. But in Singapore, for the last 10, 20 years, we have observed that when there's a switch in the predominant serotype, 3 to 6 months later, we get an outbreak. And we got Dengue 1 outbreak when it switched from Dengue 2 to Dengue 1 in 2004, and then 2007 back to when it switched back to Dengue 2. You can see the chart here. And very surprisingly, in more recent years, the pattern has switched. If you look at the 2019 outbreak, 2019-2020 outbreak, there was no switch in serotype, and yet there was an outbreak. But there was an increasing dominance of Dengue 3, which properly gained dominance in 2022 and caused a very big outbreak in 2022. So serotype, because it has got to do with herd immunity, it plays a very big role in whether it's going to be an outbreak or not. Today, we are going to watch, because we introduced Wolbachia, and the whole pattern now, I think it's going to change, it's going to evolve. So akan datang, I don't have the data. We need to watch for the impact of Wolbachia on all this serotype switch. But I think for many countries, it has been reported that serotype switch can be a harbinger to outbreak. Then we ask ourselves, do we have in town a high epidemic potential virus? We know that we have in our surveillance, we know there are loads of viruses in Singapore. Singapore is hyperendemic. That means we have 4 different serotypes. In each serotype, we have multiple genotype, lineages, clades, whatever you want to call it. So yeah, we have a lot of them through importation or through local in situ evolution. So which one is going to cause trouble? And we just simply plot this accumulative cases. We model it, and a very simple model, and you can see that which virus is causing problem. In 2022, we saw a Dengue 3, genotype 3 variant sort of escalating. It was shooting up at a very high speed. So that sort of really alerted us to a potential outbreak. There was a serotype switch to Dengue 3, and then you see this virus is really escalating, accelerating in speed in accumulation. And we even put the virus into the mosquito, and we saw a very short extrinsic incubation period, a chart on the right. It was very similar. The extrinsic incubation period, or epidemic, which can be translated into epidemic potential, was similar to a Dengue 2, which caused a previous big outbreak. And that really alerted us to, hey, this is not going to be an easy year. And there in 2022, we did get a big outbreak. Together with all this data, when we get all this data, we put together, and we have a spatial alert outbreak model, spatial lasso model, and we can then predict to say, we can then assess to determine whether we're going to how big our outbreak would be. So these are all very useful information for us for risk assessment, to determine the potential outbreak so that we can prepare for it. Another aspect of understanding the virus, I think associating it with diseases is very important. This data is obtained from our United Dengue program. very nice data from Malaysia. If you look at the dengue 1, if you take dengue 1, genotype 1 as a reference point, then genotype seems to be less severe. All right, then Den 2 we know often is more severe and is similar to dengue 1, genotype 1. But if you look at Den 3, genotype 3, and Den 4, they are milder. And I think that is something that a lot of people may sort of, clinicians may actually feel it already. You have a gut feel. But this is one set of data, and I think we really need to study the virus together with severity and together with epidemiology to understand what the virus can do. Today, very little knowledge, so it's very hard to predict whether we have a nasty virus in town. This is one other function for us to do genomic analysis is with the geophylogenetic analysis. You could actually see connectivity between cities and between places. And here, in fact, you can understand major hubs. And in terms of in terms of prioritization, then you can programs then can target on all these hubs. So we can, in fact, our Wolbachia was done this way. We did this phylogeo analysis to understand where the hubs are, and there we select these places for prioritization. Associated with the slide above just now, this is a paper again under United Dengue that we published in 2013. There was a dengue 2 virus that was higher fatality compared to other parts of Malaysia. So again, this one alerted us that hey, there could be a nasty virus around. So important, the power of phylogenetic analysis of dengue virus, it's important. This is also under United Dengue. Our network is our program where we share data and help each other out in the region. This is in the Philippines. Again, you can see from how the virus travel by genetics and by time. You can see where the hub is and where we could target. There's limited resources. I'm going to switch tone because I think chikungunya had some very good success when it comes to using genetics. If you remember 2000, in the mid-2000, there was chikungunya outbreak coming up from, I think it started more or less from Kenya region and then La Reunion and then came to Asia. And there was in fact, the whole process, there was mutation, what we call the A226 mutation. And that mutation has allowed the virus to adapt to Albopictus, Aedes albopictus. Classically, chikungunya is transmitted by Egypti, which is more an indoor mosquito. But this mutation caused it to adapt to Albopictus. We want to thank the French team and the US team that identified this mutation. So when it came to Singapore, when we sequenced it and we realized, hey, we shouldn't be just fighting Aedes aegypti. We now have to fight Albopictus too, which is outdoor. So we did a lot of fogging outdoor. So that's again the importance of understanding the genetic. It can really help us understand our epidemic. If we put epidemiology, virus, competency, and severity together. We also showed that in our years of sequencing, we have 4 distinct groups of chikungunya virus, the African genotype, circulating in Singapore. But in fact, when we put all these data together, we realized that we are not endemic for chikungunya. We were worried, when we see a chikungunya case, it's like, hey, was it from last year has there been a silent transmission? And from all this data, we showed that there's no silent transmission. In fact, there are transient transmission, importation, small transmission, and then so each of the transmission each time was a different virus. So that gave us the assurance that our vector surveillance and control measures were effective in averting a severe epidemic. Right, so this is again part of our effort using genetics to do risk assessment to evaluate our situation. And this data is corroborated by our seroprevalence data, showing just 1 to 5% seroprevalence with IgG. The population, only 1 to 5% has the IgG. Same thing for Zika. Recently, we have a few Zika cases. Again, in 2016, we had an outbreak, and then 2023, 24, and each time they were with different viruses. Again, it tells us that it gives us a sort of confidence that it wasn't a continuous transmission. They were transient and they were mitigated, and then new viruses came in. I will end here. In short, genomic of viruses, dengue viruses is important for epidemic potential, to understand severity, and to help us to do risk assessment, whether it's a serotype switch telling you there's an outbreak, or genotype looking at lineages to tell you what your situation is like, whether you're very endemic, whether you have multiple lineages around, or is it just sporadic clusters that you mitigate each time. Okay, I will end here. Thank you.
Many thanks, Professor Litin. We are running a few minutes late. I will take one question for the presenter. So this is for Nuno. Nuno, there is a general initiative to reclassify dengue virus serotypes to distinct species in the broader dengue clade due to genomic diversity between them. How does this community view that goal in terms of relevance to vaccine initiatives, epidemiology, diagnostics, assays, and awareness?
I cannot really thank you for the question, but I cannot really respond for the whole community, of course, right? My understanding is that yes, there have been different initiatives to reclassify dengue, not just as different species, the different serotypes, but also there have been different initiatives to classify distinct genotypes differently as well. And I think it would be very useful to have a standardized way that everyone around the world sequencing and seeing dengue cases and sequencing those cases uses, so we can directly compare dynamics across different regions.
Thanks, Nuno. And to Dr. Gamu, can a patient have co-infection of dengue and yellow fever?
Yes, it's possible if it's an area with both viruses are co-circulating with the presence of Aedes aegypti. I haven't seen yet such a case, but last year we had a co-infection in the south of the country between dengue and chikungunya. Thank you.
Thanks. And to Solitin, it is possible that when using Wolbachia to control Aedes mosquitoes could push faster mutation of dengue serotypes for them to cope with Wolbachia?
So in Singapore, there are two ways of using Wolbachia. One is to replace the population with mosquito with Wolbachia. Another strategy that you could use is that you release only male mosquito, and when a male mates with a female out there that has no Wolbachia, you get suppression of mosquito population. So when you have suppression of mosquito population, it's like mosquito control. We don't see any adaptation unless that you could get viruses with higher epidemic potential. Even a few mosquitoes, they are really super good. Within 1 day, they can start with a short extrinsic incubation period. But it's not the adaptation risk is not associated with the suppression method, but the current hypothesis that it could happen in the replacement method, but that is not proven yet. So I think we need to watch. Thank you.
Thanks very much. And thank very much to the presenters and for all the interested in the audience. We have many questions. We will be answering some of the chat, and I will give now the word to Dr. Diana Rojas for the closing remarks. Thanks, everyone.
Thank you very much, Josefina. And let me start by thanking all the invited speakers for the very informative presentation, and also for the participants for connecting and actively participating on this webinar. I also want to thank the International Pathogen Surveillance Network for the collaboration to strengthen the global network of arbovirus genomic surveillance, where we will together establish a global landscape of Aedes-borne virus genomic surveillance, establishing a WHO pipeline to track the diversity of these arboviruses, and also conduct a global survey on laboratory capacities for genomic surveillance of arboviruses. So this is a great way to kickstart this collaboration with the IPSN. So as you hear, during 2024 and the current dengue global emergency, almost 1.5 million cases have been reported. And with the support of the EPI-WIN team, we have been hosting several updates on the dengue global situation. So also thank you to the EPI-WIN team. So as I know, we are running out of time. Looking forward, we want to invite you to the upcoming WHO Public Health Laboratory Knowledge Sharing webinar series, which will be held on the 11th of September of 2024. The topic will be dengue virus testing. We had a lot of questions about dengue testing, ELISAs, RT-PCR. So this is the right webinar to join. And so it will be also in the context of the global outbreak and aligns really well to the topic that we just covered in this EPI-WIN. So thanks to all, and see you on the next EPI-WIN. Have a great afternoon, morning, or evening. Thank you.
Okay.
Thank you.
Thank you very much to Diana and to the entire panel for a wonderful webinar. Before we leave, I know we've run out of time, but very quickly, we have one more Slido where we would like to hear from you. The key messages, just one. So we would like to hear from you. What, in less than 5 words, what is your key takeaway from this webinar? What is it that you found most interesting or most effective? Just very quickly. I request my colleague to share the screen so we have a very quick idea. The slide, go to slido.com. Just very quick feedback from you, because this is your webinar and we'd like to know what you found most useful. Great, it's great to see the responses come in and to see what you found useful. And we look, as Diana mentioned, we look forward to having you in future webinars. It's great to have such enthusiastic people in this journey that we're taking together through EPI-WIN. And to let you know too that in September, and we will let you know closer to the time, we intend to focus on Mpox, different aspects of Mpox as well. We're trying to organize and fix the dates with our experts, and we will let you know through our usual channels what the webinars are. And we will, of course, again, work with our colleagues in the arbovirus team, thanks to Diana, who I know most of you are already familiar with, and to Josefina, and also thanks to other colleagues who are in the background, like Dr. Homa Attar, Ingrid Rabe, Lorenzo Subizzi, my colleague Sadrish Pradhan from the EPI-WIN team, the Project ECHO team, the interpreters, the French and Spanish interpreters, and the IT support who've been working very hard in the background. Thanks to all of you, and thanks to all the participants. And I think it's great. We see dengue surveillance as a key takeaway and genomic surveillance, and a few others, molecular epidemiology, collaboration, but genomic surveillance, virus surveillance, Wolbachia, were some of the keywords that we're picking up right now. Thank you very much with that, and look forward to the next webinar. Thank you very much to the panelists and to all the speakers.
Thank you.
Bye-bye.
Thank you very much.
All right.
Thank you very much. Bye-bye.
Thank you.